All right, so in the previous chapter, we talked about passive transport. In this chapter, we are going to talk about active transport, which is going to be used to move things against the concentration gradient. And because of that, it's going to require some sort of energy to be used. In most cases, that energy comes from a molecule called ATP. We've talked a little bit about ATP already, but we will talk a lot more about it and how it's made in the coming chapters. ATP is a nucleotide, and it has 3 phosphates on it. Each of these phosphates contains a bond here, and that bond is stored energy that can be used to do work. So we're going to talk about electrochemical gradients first. Then we'll talk about primary active transport versus secondary active transport here. So we talked a bit about gradients previously, but we have this added electro part to electrochemical gradients. Not super important for us right now, but if you take anatomy and physiology, the electro part will be very important because it's involved in many processes, nerve impulses, things like this. The chemical gradient, we will definitely talk a lot about here. We are talking about the difference of ions on one side versus another side of a membrane. So having a different amount of ions inside or outside of the cell, essentially, can build a gradient. We could have something like in a typical cell where we have a net negative charge on the inside of the cell and a net positive charge on the outside of the cell. Like in this example, we have sodium that is being pulled by its gradient into the cell, but we also have potassium that wants to go out by the gradient. And these forces are part of electrochemical gradients, right? So they will kind of interact with one another, but also they'll move on their own. We're going to focus on the gradient part, not the electro part here. So these gradients, we might want to work against them. Like say, I don't know, we want to bring more potassium into the cell, right? That's something we might want to do. Maybe we want to keep the potassium inside. Well, we're going to have to go against this concentration gradient here. Or vice versa, right? If we wanted to bring this sodium out of the cell, so on and so forth, we're going to have to go against a gradient. There are 2 ways that this can happen. With primary active transport, we can directly move these things using ATP to go against the gradient. Or we can do this secondary active transport that doesn't directly use ATP, but might use an electrochemical gradient. So let's say we could be using this potassium gradient to also help pull sodium outside the cell or pull chloride outside the cell, something like that. That would be an example of secondary active transport. So we'll see some examples of primary active transport and then secondary active transport here. So why do this? Well, cells often want to stockpile stuff, right? There is very limited resources outside of the cell. They want to bring it inside and keep it inside. So they will spend energy in the form of ATP to pump molecules inside of their cell. That's going to take up a lot of energy, and the cell is going to need to maintain this gradient for it to continue living. So here we have these green molecules outside, and we want to put them inside, but we already have a high concentration inside of the cell. To move them in, we have to use a pump that requires ATP to move them into the cell. One example of this might be how sucrose can be transported across a membrane. So sucrose is a disaccharide, right? And usually there's less sucrose outside and you want to pump it inside, right? You want to get as much sucrose into your cell. So as you're bringing the sucrose in, you're building a high concentration inside your cell. By diffusion, it would want to move back out of the cell, but we want to keep bringing it in, even though there's a high concentration. To do that, we need energy. And I've told you that we could use ATP, but there's a second way we could do this. We could use another gradient to move things with it. So in this case, we have a bunch of hydrogen ions that are at a high concentration outside of the cell, and they will flow through this, what we call a co-transporter, which will move the hydrogen ions, allowing them to go through. And in the same time, will pull sucrose through into the cell, pumping it to a higher concentration. That's secondary active transport. Now, of course, this hydrogen ion gradient was created through a proton pump. These are protons. H plus is a proton. We've spent ATP over here. So to actually move the protons, the hydrogen ions, we've spent ATP. That's primary active transport. But then this is secondary active transport to bring the sucrose in. There are a number of proteins that can carry different things for active transport. We classify them based on how many things they carry and what direction they're going. So we have things like A here, which is a uniporter. That means it moves one substance, okay, and it will move it by active transport. On the other hand, this one here moves both A and B in the same direction. We would call that a Symporter, Sim being the same, so it's moving two things in the same direction. Finally, we could have an example where we have an antiporter, where A moves in and at the same time it pulls B out of the cell, so moving two things in opposite directions. Okay, here we have an example, right? Hydrogen ions flow through and pull sucrose along in the same direction, so that would be a Symporter here. Let's take a look at another example here. So what type of transporter is this? We have sodium at a high concentration outside of the cell, and we have potassium, the little orange egg-like shaped things, at a high concentration inside of the cell. You can see sodium is being moved outwards, and our potassium is then being pulled inwards. So what type of transporter is this? That would be an antiporter, right? We have one thing being pumped one way and the other thing being pumped the other way. That's antiporter. This example is the sodium-potassium ATPase antiporter pump. Okay, it's called an ATPase because it takes ATP and uses the energy from one of those phosphates to power the pumping of sodium, and then in the process, as it goes back to its normal state, the potassium is pulled into the cell. And you can see we're pumping against the gradient in both directions. We're moving it out to a higher concentration, and we're moving the potassium in to a higher concentration. It doesn't actually do this in equal amounts. 3 sodiums go out, 2 potassiums come in for every 1 ATP being used. This allows the electrochemical gradient to be maintained, right? All of this relies on the 3D shape of the protein and the change that's happening. So here, right, it's at a certain shape, and then we add the ATP energy, which changes its shape to pump those sodiums. And then that shape allows the potassium to bind to it, and it falls back to its normal state, and then it pumps it into the cell. Another example of this is the sodium glucose transporter. This one is very important if you've ever drunk a sports drink, okay? So something like Gatorade or Tailwind sports drinks. They have a large amount of glucose in them. Part of that's for energy, but the other thing they have is sodium, which is an important electrolyte when you're doing physical activity. Particularly long distance running, you tend to lose a lot of sodium, so you need to replenish that. The way that we move sodium from outside of our cell into our cell is using this glucose sodium transporter. So you can see over here, we have a sodium potassium pump, right? That's our antiporter. We build up the sodium gradient outside of the cell, and then we want to move the glucose into the cell. So to do that, the sodium binds to this and the glucose binds to this. The sodium gradient helps us pump the sugar inside our cell. So the salt in your sports drink is helping pull the sugar into your cell where it's needed to then build energy from that. So our sodium ion gradient is powering this glucose pump here. To build that gradient, we have to use a different pump, right? This is the process to get glucose into our cell. Many amino acids are also transported in a similar mechanism to this. All right, let's review before we finish this chapter here. So where would an active transport protein embedded in the membrane have been made in the cell? So we have this proton pump here, and you can see it is a transmembrane protein. It goes across it. It's part of the embedded in the membrane. Where would that have been made to have this be the case? Go ahead and pause the video. Think about that. All right, this protein would have been made in the rough endoplasmic reticulum. Remember, rough endoplasmic reticulum has the ribosomes on it, so it'll make proteins, and those proteins are often embedded in the membrane. So they're in membrane from the ER, they move to the Golgi, and then finally to the cell membrane where they stay. All right, so we talked about active transport. Active transport uses energy to move substances against the concentration gradient. Human cells normally keep an electrochemical gradient of ions, which both have a concentration difference and a charge difference. We're not too worried about the charge difference here. The concentration one is the one we're going to focus on. In anatomy and physiology, you'll talk about the electrical part of it. Primary active transport directly uses ATP to move things against the concentration gradient, whereas secondary active transport does not directly use ATP. Instead, it uses another electrochemical gradient to move things, like our proton gradient in mitochondria that we will see in the next chapter. This is an important example of this secondary active transport. We talked about 3 different types of transporters. Uniporters move things in one direction against the concentration gradient. Symporters move 2 things in the same direction, and antiporters move 2 things in the opposite directions. All right, that's it for active transport. In the next section, we will talk about moving really big stuff into and out of the cell.